I wanted to thank the ARCS committee for the opportunity to share my research today. My research is centered around characterizing nutrient acquisition pathways utilized by pathogenic bacteria in order to thrive in the host environment. The heart of my research lies in enzyme biochemistry, and furthering our understanding of bacterial biochemistry will provide the impetus for novel therapeutic breakthroughs. And so with that, let's head to the lab to learn a little more about how this work is done.
Oh, hey, I didn't see you there. My name's Lindsay Matthew. I work in the Lab. Part of my research focuses on the isolation of these bioactive compounds from pathogenic bacteria. And due to the sensitivity of these compounds, oftentimes requires strict environments, oxygen-free. Let's talk about how and why this is important.
So the typical red blood cell contains roughly 1 billion heme molecules per cell. Now, importantly, heme is required for oxygen transport all across the body, and the real rock star of this story is the iron at the center, responsible for transport. Now, if we were to consider bacterial pathogens that invade the body, oftentimes they require iron as an essential nutrient for virulence and survival. Now, in an oxygen-rich environment, several of these pathogens and their respective pathways have been characterized, including Staph aureus, Mycobacterium tuberculosis, and Neisseria meningitidis. But if we were to stop and consider an anaerobic pathway or an oxygen-free environment, such as the one experienced by E. coli in the distal portion of the intestine, this environment has not really been so carefully characterized. And recent work in our lab has looked at heme degradation to this new tetrapyrrole termed anaerobilin, successively liberating the iron molecule from the center. And so part one of my project, which I've already talked about, is the isolation of this tetrapyrrole. This is goal one. But goal two will focus on characterizing these reaction intermediates that get us from point A, heme, to point B, anaerobilin.
The third goal of my research is to determine the crystal structure of QW, the enzyme responsible for heme degradation in pathogenic E. coli. Structure informs function. And so by determining the structure of QW, this will inform our understanding of the mechanism for heme degradation. It is this constant battle between host and pathogen for resources that drives the evolution of these pathways. And by understanding the intricacies of these pathways, we will further the field of enzymology, but also pave the way for the development of new therapeutics.